For today, our discussion will be about the history and the basic concepts that you should know about developmental biology. To start off, our objectives are as follows. First is to define the study of developmental biology. Second is to compare it with embryology. Third is to identify the different approaches in the dev bio in other fields. Next is to identify or define terminologies for you to be able to understand concepts in dev bio. Next is to trace the lineage of early embryologists or biologists with their contributions. And of course, we will also check on the different model organisms being used in dev bio and its importance in understanding defects, may it be in humans or animals. To compare and contrast embryology versus developmental biology, it is always good to remember that most organisms do not automatically develop in full form. So there is always this relatively slow process of progressive change that we call development. And in nearly all cases, the development of a multicellular organism begins with a single cell we call the fertilized egg or the zygote. So this zygote divides mitotically to produce all the cells of the body. Hence, with embryology, it is the traditional terminology used for studying animal development that exists only between the phase of fertilization and birth. It also focuses on observational biology, measuring characteristics with or without experimental manipulations. However, most organisms never stop developing. Just for example, the replacement of your skin cells. So from time to time, we have this capability of regenerating skin cells. Another example will be our bone marrow, which actually sustains the development of our new red blood cells. And of course, in some animals and plants, they also have the capability of regenerating their severed parts. So in recent years, it has been customary to adapt the developmental biology as a discipline that studies embryonic and other developmental processes, and it expands embryological studies using molecular techniques and genetic approaches to study the different model systems. So again, with embryology, more of the studies being dealt into it are those phases in between the fertilization stage up to the birth stage, and then it only uses characteristics or observational observational characteristics. But with developmental biology, we try to integrate molecular techniques and genetic approaches for us to be able to trace the embryonic development from the beginning until the end of its development. So with development, it actually accomplishes two major objectives. The first one is to generate cellular diversity and order within an individual or organism, and it ensures the continuity of life from one generation to the next. But then there are also fundamental questions in dev bio that should be addressed, and one of them would be, how does the fertilized egg give rise to the adult body, and that how this adult body produces yet another body. So these are just fundamental questions that lingers about developmental biology, and there are several categories that we can use to scrutinize such questions. So the first one would be about differentiation. So with differentiation, it is actually the generation of cellular diversity wherein a single cell or a fertilized egg can give rise to hundreds of different types of cell. So basically, in the beginning, these types of cell are very generalized in characteristics. But then as development goes, these certain generalized cells are developing into specific types of cell. So it can be a muscle cell, an epidermal cell, it can be your neurons or maybe your blood cells and many more. Okay. So the second one is about morphogenesis. Since with morphogenesis, this is actually the creation of ordered form that involves coordinating cell growth, cell migration, and cell death. So next, you also have what you call the growth. So this is defined as an irreversible constant increase in the size of an organ or even an individual cell during the life history of an organism. Next is about reproduction. So it is a biological process by which an organism reproduces an offspring who is biologically like the same organism. Next is about regeneration. So this is the ability of some organisms to regenerate every part of their bodies. Next is the environmental integration. So with environmental integration, these are cues, environmental cues that influences the development of organisms. And then next, you have the evolution, which involves the inherited changes of development. So developmental biology, according to Love and Minelli, is the science that investigates how a variety of interacting processes generate an organism's heterogeneous shapes, size, and structural features that arise on the trajectory from embryo to adult, or more generally throughout a life cycle. So basically, developmental biology is a multi-field study. So with this, you can study about the anatomical changes in an organism. You can also check on the physiological processes in one's body system. You can also check on the molecular biology or the DNA of these organisms. And basically, these developmental biology studies are very useful when it comes to cancer research, neurobiology, immunology, and of course, understanding the evolutionary changes of each organism. So in developmental biology over the years, there were model organisms that have been used in order to understand developmental changes depending on what type of species. So for example, we have the Sinoradites elegans, which is a type of nematode. They have also utilized the Drosophila melanogaster or the fruit fly. Some also used sea urchins. Some also use the Sinupus laevis or the South African claw-toed frog. Some also use a different species, which is the Rana pipiens. And then some also use the Gallus gallus, specifically the use of the chick embryo. Some developmental studies also use duck embryo, so which is what species is more likely to be available, they try to use it. Okay, since the development of chick embryo is just lateral to the duck embryo. It's just that they come from two different species of birds. Okay, and then you also have the mus musculus or the lab mouse. And then for plants, they have utilized what you call the Arabidopsis thaliana, which is a type of an angiosperm or a flowering plant. So for us to further understand the history and the background of how developmental biology succeeded up until to this time, we will now be checking on some of the early biologists and philosophers that have contributed to this study. So first and foremost, we have Hippocrates. So he started a scientific approach to explain the development in Greece in the 5th century BC. So basically, he explained development in terms of the principles of heat, wetness, and solidification. However, of course, not until such the studies in sciences have progressed, the approaches in understanding developmental biology has been changed and of course has been upgraded. Okay, so initially, everything um, in understanding history and, of course, evolutionary changes, we have dealt into studying anatomical and or descriptive studies. So even in the um, initial years of, for example, discovering plants or animals, all since there's no softwares yet, or maybe um, high technology devices that we could use in tracing such development, philosophers and even scientists or biologists has just used their naked eye to be able to describe and to be able to annotate different organisms that they have studied. So more likely, they just based on the features of such animals and of course the anatomical changes and describe them in different writings. And then, of course, there has come a time that um, some of the scientists already used experimental approaches to be able to understand different effects of manipulating development. And then, of course, in these recent years or the more modern years of understanding development, we have utilized genetic studies. Okay? So, for the early biologists that used anatomical or descriptive studies, one of them was actually Aristotle. Okay? So, if Hippocrates started his theories about development in the 5th BC. So it's around siguro 1 century pa after him that Aristotle, a Greek philosopher, addressed the problem of how the different parts of the embryo were formed from two possibilities. So the first one is about everything in the embryo was preformed from the very beginning and simply got bigger during the development. And then the second one is that the new structures arouse progressively, a process that he termed epigenesis, which means upon formation and likened metaphorically to be as knitting of a net. So this is he actually was also the first one to figure out the functions of the mammalian placenta and the umbilical cord. So his writings of saying that embryo has been preformed, of course, in those century or the years that he's very famous and he's well known, so more of the scientists were actually believing about his theories and his explanations. Okay? So in comparative embryology, so this is an application about Aristotle's work. So in his generation of animals, he noted some of the variations on the life cycle theme. So it is more on the fertilized egg. So how did he um, describe the fertilized egg by using descriptive or anatomical studies? He said that some animals are born from eggs, which we call the oviparity. So examples of it would be the birds, the frogs, and most invertebrates. So he also categorized some of the fertilized egg as viviparity, or those animals that are born by live birth. So those are the placental mammals or animals. And then next is the ovoviviparity, wherein some produce an egg that hatches inside the body. And some of it, or some of the examples would be certain reptiles and the sharks. Okay? So those are his contributions when it comes to comparative embryology of the fertilized eggs. So he also presented the major cell division patterns by which an embryo is formed. So the first type of this pattern is what you call the holoblastic pattern. So this is when the entire egg is divided into successively smaller cells, like what you see in the development of frogs and mammals. And then you also have the meroblastic pattern, wherein only part of the egg is destined to become the embryo, while the other portion, or the yolk part, serves actually as the nutrition for the embryo, as in chicks. So with these types of animals, the yolk serves to be like their food up until they are able to hatch their egg and be able to feed themselves. Okay? So next would be William Harvey. So in 1651, he concluded that all animals, even mammals, originate from eggs, which he wrote in the On the Generations of Living Creature, with the motto ex ovo omnia, meaning all from egg. So he's already generalizing that, may it be reptiles, birds, humans, or insects, they all come from eggs. Okay? So he is also the first one to see the blastoderm of the chick embryo. He's also the first one to notice that there are islands of blood tissue that form before the heart does. So this islands of blood tissue, it can be the red blood cells or the white blood cells that he found in the blood. And then he also suggested that amniotic fluid might function as a shock absorber for the embryo. Next, we also have Marcello Malpighi. So he is an Italian embryologist, and in the 1672, he published the first microscopic account of chick development. So he first identified the neural groove, which is actually the precursor of the neural tube that eventually becomes your backbone or your spinal cord. Okay? So this is also the muscle forming somites and the first circulation of the arteries and veins to and from the yolk. So that's his extensive study now of the cheek development, pertaining to more on the nervous system side of this development. Okay? And then he began one of the greatest debates in embryology, which is the Preformationism. So remember that Aristotle um, opened the idea of epigenesis, but then Marcello Malpighi um, gave his different view about development, which is about preformationism. So when you say preformationism, it showed here on his experiment that you see on the screen right now that the unincubated chick egg already had a great deal of structure, and this observation provided him with reasons to question epigenesis and advocate that preformationist view, according to which all of the organs of the adult were prefigured in miniature within the sperm or the egg. Okay, so ito naman, it says that it is already prefigured in within the sperm or the egg. Okay, so here organisms were not seen to be constructed but rather unrolled or unfurled. So he believes that coming from the sperm or the egg, this organs are already there. Naka-roll lang sila and ina-unroll lang sila kapag ka nagde-develop na. Compared to the idea of epigenesis. Okay? So, epigenesis versus preformationism. So again, epigenesis is upon formation. So this is a theory of development that states that the new structures arise by progressing through several different stages. On the other hand, preformation theory suggests that all structures exist from the very beginning and they just get larger. Okay? So, again, the epigenesis, it follows a certain process, but then again, in preformation, naniniwala sila na nandun na yung structure na yon at lumalaki na lang. Okay? So, one preformation theory that has been formulated during that time is actually the theory of the homunculus. So it suggested that a little human embryo was hidden in the head of every sperm. So in this drawing that you're seeing right now, it is actually an imaginative drawing after Nicholas Hartsoker. So, yan na there is a homunculus that was curled up in the head of each sperm. So, yun yung sinasabi kanina ni Malpighi na it's either on the egg or the sperm na meron na talagang structure. Okay? Na nandoon sa type of cell na yon na lumalaki na lang sila eventually. But then again, with Aristotle, with the idea of epigenesis, he believes that the development of new structures comes from different several stages. Okay, so this was one of the most interesting debates that have concurred in studying developmental biology. So next we have Caspar Friedrich Wolff. So he observed that the development of the chick embryos as well. He demonstrated that embryonic parts develop from tissues that have no counterpart in the adult organism, thus supported epigenesis. So, ibig sabihin, his findings actually supported the development or the idea of epigenesis rather than the preformationism. Okay? Because, tama nga naman na kung iisipin mo, kapag ka ang sinunod mo ay kay Malpighi, na sinasabi niya na meron ng mga structures dun sa sperm or sa egg at lumalaki na lang sa eventually, bakit hindi nakikita dun sa mga chick embryo na inaral niya yung same part na meron dun sa egg na yon compared to the adult organism? Okay? So, for example, sabihin natin, kung sinasabing chick embryo, ano ba ang meron sa chick embryo? So, sabihin natin yung um, wings, okay? So, sa egg ba nakita yung wings? Hindi. Pero nakikita ang wings lang pagdating sa adult type of the organism. So, yun yung support of the idea of epigenesis. So, with Wolff, more likely na nakaside siya dun sa idea of epigenesis. And then, additionally, he also postulated an unknown force, which is the V essentialis or the essential force, which, acting according to the natural laws analogous to those such as gravity or magnetism, would organize embryonic development. So, siya naman, he tried to, other than the idea of epigenesis, he also pitched his idea that there's a certain force that affects embryonic development, which is more or more known of these forces are the gravity and magnetism. So parang nag-apply siya dito ng physics. Okay? Na may kinalaman ang gravity at magnetism sa pagbubuo ng mga embryonic development. So next we have Immanuel Kant. So he is a German philosopher. He tried to reconciliate preformationism and epigenesis with his colleague Johann Friedrich Blumenbach. So they postulated, or he postulated, a mechanical goal-directed force he called Bildungsstrebe, or the developmental force, which was not theoretical but could be shown to exist by experimentation. Okay, so these partners, so they tried to combine the idea of Aristotle and Malpighi. So next, we have Heinz Christian Pander. So he is a Russian biologist. He visualized and discovered epigenesis of the 3 embryonic germ layers. So this is one also of the most important events in developmental biology. So when you say the embryonic germ layers, this is the ectoderm, mesoderm, and endoderm. And he was also the first one to introduce the first indication of induction, where no vertebrate tissue is able to construct organs by itself, and it must interact with other tissues. So again, with the development of tissues in the body, he is saying that kailangan nakikipag-cooperate yung mga cells or tissues na yun with other cells found in the body to be able to produce another tissue. So next we have Carl Ernst von Baer. He actually was the first one to describe the ovum in ovarian follicles of a dog or the mammalian egg. He also described the notochord as a distinct and primary structure. And he actually repeated Pander's investigation on chick. And then he proposed 4 laws of development. The first one is that the general features of a large group of animals appear earlier in development than do the specialized features of a smaller group. And then next is less general characters develop from the more general until finally the most specialized appear. And then next, the embryo of a given species, instead of passing through the adult stages of lower animals, departs more and more from them. And therefore, the early embryo of a higher animal is never like a lower animal, but only likes its early embryo. So, from his 4 laws, it's already giving you the idea na kahit papano nabubuo na yung concept of how really the development of um, how a development of an organism happens. So, nagkakaroon na ng, kumbaga, laman yung theories of, or combined theories of this early biologist. So, from here, he is already believing that nagkakaparehas lang ang higher animals at lower animals during its early embryonic period. Okay? Next, we have Martin Heinrich Rathke. So, in here, he pioneered studies of vertebrates and crayfishes. So he was the one to produce important monographs on the development of fish, reptiles, and vertebrates in general. He focused more on the details of individual transformation of these different organisms, followed by the development of vertebrate skull, the excretory system, and respiratory system. He is also first to identify the pharyngeal arches, and he showed that the same embryonic structure that became gill support in fishes became jaws and ears in animals. So in here, you will see that there is already, again, a comparative anatomy concept. No? Kasi from those embryonic structures that he saw from fishes, he compared it with the jaws of and ears of other animals. So, the cell theory actually changed the concepts of embryonic development and heredity. So, how? So, how important is this cell theory? So, in 1665, Robert Hooke published Micrographia with drawings and descriptions of organisms that he viewed under his invented microscope, leading to the discovery of cells. So, this led to the development of the classical cell theory. So in 1839, the first 2 parts of the classical cell theory was proposed by Theodor Schwann based on his conclusion with Matthias Schleiden. So the first 2 parts coming from the classical cell theory states that all organisms are made up of cells, and the second one states that cells are the basic unit of life. So the third part of the classical cell theory was actually described by Rudolf Virchow in 1858 when he stated omnis cellulae cellula, or all cells come from cells, which gave them the third part of the classical theory that states that cell comes from pre-existing cells that have multiplied. So collectively, these 3 parts or these 3 statements comprise the classical cell theory that you should always remember. Okay? So, with this different changes or development that has happened and theories and laws that were formulated all through the years in studying developmental biology, one of the major triumphs of the descriptive embryology process of study is that the generalized life cycle or the processes of development. Okay? So in this generalized life cycle, the first point or the first process is what we call the fertilization stage. So it involves the fusion of the mature sex cells. So when you say sex cells, this includes your sperm and the egg, which are collectively known as your gametes. And then the fusion of the gamete cells stimulates the egg to begin development and initiate a new individual. And then subsequently, the fusion of the gamete nuclei called pronuclei from the male and female gives now the embryo genome. Okay? So next important process in life cycle is the cleavage process or the cleavage phase. So it actually immediately happens after fertilization, where there is an extreme series of rapid mitotic divisions. And this is when the volume of the zygote cytoplasm is divided into numerous cells called your blastomeres. And then at the end of this cleavage stage, it results to the formation of a sphere called a blastula. So coming from zygote, eventually it will become a blastula at the end of the cleavage phase. Okay? So pattern of embryonic cleavage is determined by two major parameters. First one is the amount and distribution of yolk protein within the cytoplasm, which actually determines where cleavage can occur and the relative sizes of the blastomeres. And then the second one is or are the factors in the egg cytoplasm that influence the angle of mitotic spindle and the timing of its formation. So, in general, yolk actually inhibits cleavage. So, if in any case that a fertilized egg does have a yolk, um, it actually inhibits the cleavage process because again, the basic function of the yolk is for the nourishment of the zygote, right? So, when one pole of the egg is relatively yolk-free, when you say yolk-free, doesn't have any yolk on it, cellular division occurs there at a faster rate than the opposite pole. So, in an egg, when you see a yolk-rich pole, it's what you call the vegetal pole. And then the animal pole, on the other hand, is actually does have a relatively low yolk concentration, and this is where the zygote nucleus is displayed. In this figure, you will see the summary of the main patterns of cleavage. So, the different classification of cleavage types are as follows. So, the first one would be the holoblastic or complete cleavage, meaning that the cleavage furrow extends through the entire egg and shows the influence of yolk on cleavage as symmetrical and has a pattern. So, kumbaga, pantay, okay? Or evenly distributed yung yolk nila in either poles. So, first type of um, isolecital type of yolk would be the radial cleavage, which are prominent in echinoderms and amphioxus. Next is the spiral cleavage for annelids, the mollusk, and flatworms. Next is the bilateral cleavage for tunicates, and then the rotational cleavage for mammals and nematodes. So as you see there, in the pictures and the drawings, it already, or you will already understand how this, or how the cleavage happens in a type of egg. Okay? Can be radial, spiral, bilateral, and rotational. Next type would be your mesolecital. So these are eggs which has moderate vegetal yolk disposition. So kung ang isolecital ay evenly distributed yung yolk nila, sa mesolecital may moderate number of or volume of yolk sa mga eggs na to. So again, kapag ka mas marami ang yolk, of course, mas lessened or mas konti yung pagki-cleave nung egg na yon. So compared to the isolecital, tignan nyo yung pictures or yung drawings dito sa mesolecital, medyo hindi siya kasing prominent ng cleavage ng mga isolecital na eggs. Okay? So, in mesolecital, the type that you can see is a displaced radial cleavage, which is prominent with amphibians. Okay? So, next, we have the meroblastic or incomplete cleavage naman. So, type would be telolecital. So, that is by having dense yolk throughout most of the cell. So, as you see here, mas kumukonte or mas nagiging mababaw yung cleavage. Okay? So, you have there the bilateral cleavage which are found in cephalopod mollusks. And then you also have the discoidal cleavage which are found in fish, reptiles, and birds. So if you notice, there's just a part of the egg that's having the cleavage, okay, for the discoidal ones. And then next is the centrolecital. So ibig sabihin, the yolk is in the center of the egg. So sabi nga natin, kapag merong animal and vegetal pole, di ba, there's just one side where the yolk is, but for centrolecital, the yolk itself is at the middle. So, it can be, kumbaga, parang nasa gitna siya ng vegetal and your animal pole. So, what happens with them is they happen to have the superficial cleavage, which is found in most of the insects. Okay. So next phase in the generalized life cycle would be gastrulation. So it happens when the mitotic division slows down and the blastomeres undergo movements and changes in their positions relative to one another. So in here, the embryo is now said to be in gastrula stage. So kanina zygote, napunta tayo sa blastula, ngayon naman gastrula na siya. So the result of these processes is the rise of the three germ layers that will interact to generate the organs of the body. So the first one would be the endoderm, and then you have the ectoderm, and then the mesoderm. So for the different types of cell movements, so this is one of the most important things also that you should know, especially in the process of gastrulation. So these movements are as follows. So you have the invagination. So it means the infolding of a sheet of epithelial cells, much like the indention of a soft rubber ball when it is poked. So invagination, so ibig sabihin papasok. For example, the gastrula of a sea urchin endoderm. And then next, you also have your involution. So this is the inward movement of an expanding outer layer so that it spreads over the internal surface of the remaining external cells. So as you see here, the example would be an amphibian mesoderm. And then next, you have the ingression. This is the migration of individual cells from the surface into the embryo's interior. So the individual cells become mesenchymal and migrate independently. So this can be found in sea urchin mesoderm and then the Drosophila neuroblast. And then next, you have the delamination. This is the splitting of one cellular sheet into two more or less parallel sheets. While on a cellular basis, it resembles ingression, the result is the formation of a new additional epithelial sheet of cells. So, as found in the hypoblast formation in birds and mammals. And then next, you have the epiboly. Epiboly is the movement of epithelial sheets, usually the ectodermal cells, spreading as a unit rather than individually to enclose deeper layers of the embryo. So this can occur by cells dividing, by cells changing their shape, or by several layers of cells intercalating into fewer layers. Often, all three mechanisms are used. So, this is basically found in the ectoderm formation in sea urchins, tunicates, and the amphibians. So, it is also important that you know the foundation of the body. Okay, so for the foundation of the body, these are just like your anatomical positioning. Okay, so you should know the anteroposterior or the AP or anteroposterior axis. So this is the line extending from the head to tail, as you see here. From head, this is the anterior side to your tail or the posterior side. Okay, so for organism that doesn't have any tails, of course, that's from mouth to your anus. Okay, and then you also have the dorsoventral or the DV or dorsoventral axis. This is the line extending from the back of the dorsum to your belly. Okay, so this one. This is the dorsal side and then the ventral side. Okay, so for humans, yung likod, yun ang dorsal, yung harap kung nasaan yung tummy, yun ang ventral side. And then of course, you also have the right-left axis that separates the two lateral sides of the body. Okay, so the lateral part, yung mga nasa sides. So anything that is going away from the midline, that's what you call the lateral sides or the left and the right axis. Okay? So next, we have what you call now your organogenesis. So this is the result of the establishment of tissues and organs from the interaction of the germ layers. So again, the germ layers, kahit na sinasabi natin na ectodermal sila, endodermal, or mesodermal, their functions are not restricted for ectodermal, for example, ectodermal derivatives. So, what happens is that pwedeng mag-work together ang ectoderm and endoderm o kaya endoderm and mesoderm to be able to produce a certain tissue. So, hindi sila independent. Actually, they are interdependent with each other to form the different organs of the body. Okay? So, pwedeng merong mga organs na solely dependent lang sila sa isang germ layer, but there are some tissues also in the body that is a product of the combination of 2 or 3 germ layers. So basically in organogenesis, we also have what you call the chemical signals, which are actually helpful in the exchange between the cells of the germ layers, resulting in the formation of specific organs at specific sites. So, ibig sabihin, these chemical signals, they are the ones responsible to pull those cells kung saan ba dapat sila naka-layout. So, for example, the cells of your head. Ano ba yung mga cells na meron sa head? So, dapat yung mga cells na yun, pumunta sila sa anterior side of your body. So, what are the cells naman found in your skin? Okay, so instead na yung cells na yun ay more on the endodermal side or the inner part of your body, usually these epithelial cells go to the ectodermal surfaces of your body or the outside part of your body. So, these chemical signals are the ones responsible for those signals. Kumbaga, sila yung mga traffic enforcers. Okay? So, they tell you where to go and para saan ka. Okay? Next is actually metamorphosis. Okay? Or kapag sinabi natin 5 processes of development, the 5th one should be growth. Okay? But then again, since an organism differs from one organism, let me tackle growth in 2 aspects. So the first one is about metamorphosis. So metamorphosis is actually the process used by organisms that hatches from the egg or those who are born not sexually mature. So in most animals, young organisms is called a larva, which can look significantly different from the adult form. And the larval stage is actually one that lasts the longest and is used for feeding or dispersal. Okay? So again, this part sa metamorphosis, it's more on the growth already for the process of development. So ayan, metamorphosis is used for those organisms that do produce larvae or larva. And then the other one is for cell differentiation. So for growth and for organisms that do not produce larva, ang nangyayari for the growth phase is more likely the cell differentiation. So we now have what you call the germ cells. So these are used to produce the next generation cells or the precursor cells. So these germ cells, ito yung mga tinatawag natin na generalized cells, no? From the beginning, sila yung, kumbaga, general cells, and then they differentiate into specialized cells as development happens. So for the germ cells, there are actually two types of cell being generated. So the first one is what you call the gametic cells. This includes the germ cells plus their precursor cells, and they produce the reproductive functions, or they are used for the reproductive functions of organisms. So when you say the gametic cells, these are the sex cells, or the sperm and the egg cells. And then the development of gametes is what we call gametogenesis. Next, the second type of cell being produced is what you call the somatic cells. So this is also known as the vegetal cells, and they give rise to the individual body. Okay, so there are only two types of cell found in an organism's body. First, it can be gametic, or the sex cells, and then the second one are all of the other cells found in your body, like skin cells, your bone cells. Those are what you call your somatic cells. Okay, so in here you see, let's have an example, okay, of a certain life cycle that wherein we use a classic example of an animal model for dead body. So that's actually a frog's life cycle of the Rana pipiens. So in here, as you see, there's the process of gametogenesis at first. So it starts with the, of course, the female oocyte and the oocyte, or the female gamete, and then the sperm that fuses and then eventually goes into the process of fertilization. So after the fertilization process, it goes to the cleavage process, wherein you will see that the zygote, or the egg, changes into a morula, and then eventually at the end of the cleavage period, it becomes now a blastula. Okay? And then when it enters the gastrulation phase, you see now the development of the blastocele, which is actually a site, okay, a hollow site within the cell. And then you will see now here an invagination for the formation of the blastopore. And then in here, at the end of the gastrulation process, you will already see the development of the three primary germ layers. So again, with the ectoderm, it's more on the outer part. Mesoderm is the inner part, and then the endoderm, this is more closely or this is more likely to produce tissues, organs or tissues in the body. Okay, and then after gastrulation, it goes now to the organogenesis, or the formation of the other organs. Okay, and then in between organogenesis and the larval stages, you will see the hatching, or the birth. Okay, so and then in the larval stages, it matures until it also produces already gonads, and then happens the metamorphosis for some species, and then maturity. So that's how the cycle goes. So once an organism is already matured or sexually capable already to reproduce, of course, the cycle of producing again the sperm and the egg would continue again another cycle. So, in a life cycle, so for example naman, we have the Xenopus laevis. Okay, this is another, or this is another type of frog species. So, the life cycle is often controlled by environmental factors. So, in most frogs, gametogenesis and fertilization are actually seasonal. Okay? Why do we say that it's seasonal? So, in most species of the frog, fertilization actually occurs external. So, ibig sabihin, fertilization is not um, ignited, or it doesn't start within the frog. Ang nangyayari, the eggs are being dispersed in water, and then yung sperm ganun din. So pwedeng sa pond doon mag-fertilize yung mga eggs. So that's what you call external. So the fertilization happens outside of the organism's body. And then they use what you call the position amplexus, wherein the male frog grabs the female's back and fertilizes the egg as the female releases them. Okay, so that's how they fertilize their eggs. So that's amplexus. So next, some species lay their eggs in ponds vegetation, and the egg jelly adheres to the plants and anchors the eggs. So the eggs of other species float into the center of the pond without any support. So in here, you see early development of the frog Xenopus laevis. So these are clutch of eggs. As you see here, there's already the animal pole and the vegetal pole. Okay. So the unfertilized egg is usually a single large cell. So this is a single cell. The animal pole is the upper part of the egg that is pigmented. And then the vegetal pole is the lower region that contains the egg. Okay? For such organisms like the frogs, since external fertilization nga sila, they would really need to have a yolk para ma-nourish sila. Kasi unlike mammals na kapag internal fertilization ang nangyayari, of course, it is the responsibility of the parent or the mother to feed their young. Okay? But for frogs, it's a different story. So they do have the yolk. And then next, of course, after fertilization, there's already the formation of the cleavage, the blastula, and the gastrula. So during cleavage, the volume of the frog stays the same, but it is divided into tens of thousands of cells. And then blastulation happens after approximately 12 cycles of division, making a layer of small cells surrounding a fluid-filled cavity you call the blastocyst that sits on the top of the large yolk eggs. So in here, you will see in letter C, or the figure in letter C is an 8-cell embryo. And then in letter D, if you zoom in it, yung mga maliliit na bilog-bilog na yan, ganyan na kadaming nag-multiply yung cells ng egg na yan. So from 8, dumadami siya. And then in letter E, it is already showing the early gastrula with the formation of the dorsal blastopore lip. Okay, so this blastopore lip, this is actually mesodermal and endodermal derivative. So combination siya. So next, you have gastrula. So the gastrulation in the frog begins at a point on the embryo surface roughly 180 degrees opposite to the point of sperm entry, with the formation of a dimple called the blastopore. So this actually marks the future dorsal side of the embryo and expands to become a ring. Okay? So if the sperm enters here sa kabilang pole, okay, the gastrulation would definitely show on the opposite side. Okay, so cells migrating through the blastopore to the embryo's interior become the mesoderm and the endoderm, and cells remaining outside become the ectoderm, and this outer layer expands to enclose the entire embryo. Next, for the organogenesis, it begins when the cells of the most dorsal region of the mesoderm condense to form a rod of cells called the notochord. So the notochord cells produce chemical signals that redirect the fate of the ectodermal cells above it, and are instructed to become cells of the nervous system. So cells change their shapes and rise up from the round body, and the embryo is now called a neurula. So once there's already the formation of notochord and the neural tube, okay, so it already depicts the start of your organogenesis. So in this picture, it's a neurula where the neural folds come together at the dorsal midline, creating a neural tube. So within that neural tube is where you see your notochord. So the neural precursor cells elongate, stretch, and fold into the embryo, forming a neural tube, and the future epidermal cells of the back cover the neural tube. So once the neural tube has formed, it and the notochord induce change in the neighboring regions, and organogenesis continues. So the mesodermal tissue adjacent to the neural tube and notochord becomes segmented into somites. Okay, so here. Para siyang mga muscle-muscle sa gilid ng notochord. Diba, nasa midline yung notochord. So, at its sides, nagkakaroon ng somites. So, these are the precursors of the frog's back muscles, the spinal vertebrae, and the dermis, or the inner portion of the skin. So, the embryo develops a mouth and an anus, and it elongates into the familiar tadpole structure. So, in here, you see that in letter G, it's a pre-hatching tadpole. So, you see here now the protrusions of the forebrain. So, these parts already parang depict the surface ectoderm for the eyes. And then, meron na din siyang stomodium or the mouth. And then, here at the parang neck area nila, you already see the gill area. And then the brain sa taas, and then of course the tail bud. So in age, this is already a mature tadpole having swum away from the egg mass and feeding independently. So the neurons make connections to the muscles and to the other neurons. So the gills form, and the larva is ready to hatch from its egg. So the hatched tadpole will feed for itself as soon as the yolk supplied by its mother is already exhausted. So ito yung mga organisms na once na na-hatch na, pinapabayaan na sila ng kanilang mother. So next is the metamorphosis and gametogenesis. So, metamorphosis of the fully aquatic tadpole larva into an adult frog that can live on land is one of the most striking transformations in all of biology. So, almost every organ is subject to modification, and the result changes in form are striking and very obvious. So, from here, the metamorphosis of a frog, there's really a huge difference, okay? So, yung mga nagkakaroon na sila ng legs, nawawala na yung tails nila, nagkakaroon na sila ng limbs, okay? So, yun yung mga, kumbaga, climax changes na nangyayari sa isang life cycle of a frog. So, B, okay, this figure is actually what you call your pre-metamorphic tadpole. C is a pro-metamorphic tadpole showing the hind limb growth. And then D is the onset of the metamorphic climax as forelimbs emerge. Okay? So E and F are already the climax stages wherein the frog is already kumbaga reaching its adult form. So, amphibian metamorphosis is initiated by hormones from the tadpole's thyroid gland. Okay, so it's very important that we remember that there are chemical signals being sent by certain organs in our body to kumbaga ignite movement in the body. Okay? So as metamorphosis ends, the development of the germ cells or the sperm and the egg begins, and for the gametogenesis to become mature, the germ cells must be competent to complete meiosis. So for the second experimental approach that we have presented earlier, apart from the traditional descriptive studies, we also have the experimental approaches. So for the early experimental approaches in DevBio, one of the known early biologist or philosopher that time is August Weismann in the 19th century. He is a German biologist. He proposed that the offspring does not inherit its characteristics from the body or the soma of the parent, but only from the germ cell, egg, and sperm, and that the germ cells are not influenced by the body that bears them. So basically, he is saying that any characteristic coming from the germ cell of the egg and the sperm, that's the only thing that's being transferred to the offspring. Okay? So he drew a fundamental distinction between germ cells and the somatic cells or the body cells. And then characteristics acquired by the body during an animal's life cannot be transmitted to the germ cells. Okay? So as you see here, there's an image about the distinction between germ cells and the somatic cells. So in each generation, germ cells give rise to both somatic cells and germ cells. Okay? So, but the inheritance is through the germ cells only. Changes that occur due to the mutation in somatic cells can be passed on to their daughter cells but does not affect the germline. So that's actually true. Okay, so again, the germ cells are only, kumbaga, they are generalized, but then again, the somatic cells, yun yung nagpo-provide daw ng, kumbaga, nagpo-provide ng change dun sa kanilang mga daughter cells or kumbaga, nagbibigay ng characteristic dun sa kanilang mga daughter cells. Okay? So, in here, there's also an explanation about the mosaic versus regulative modes of development. So, when you say mosaic model in the 1880s, Weismann put forward a model of development in which the nucleus of the zygote contains several special factors or what he called determinants. So, he proposed that as the fertilized egg underwent division or cleavage, these determinants would be distributed unequally to the daughter cells and so would control the cell's future development. So for example, in here, Weismann's nuclear determinants, if there are several determinants or factors in it, during the first cleavage, pwede daw siyang mag-jumble-jumble. Okay? So he doesn't believe that magiging na-identical dapat ang mga daughter cells. So, in the second cleavage, you see here that, kumbaga nagkaroon ng sariling partition yung mga cells. Okay? So, that's how he presented the mosaic model of development. Additionally, the fate of each cell was therefore predetermined in the egg by factors it would receive during cleavage. So the theory also assumed that early divisions must be asymmetrical so that the daughter cells would be quite different from each other as a result of an equal distribution of nuclear components. So unequal na, asymmetrical pa yung presentation ng mosaic model. So next, we have Wilhelm Roux. He is a German embryologist in the 1880s. He tried supporting the ideas of Weismann using frog embryos. So after allowing the first cleavage of the fertilized frog egg, Roux destroyed one of the two cells with a hot needle and found that the remaining cell developed into a well-formed half larva. So he concluded that the development of the frog is based on a mosaic mechanism, the cells having their character and fate determined at each cleavage. So in here, he already presented an experimental approach wherein, di ba, pag nagde-divide ang cell, one of the daughter cells are being manipulated, and then nakita pa rin niya na he can still produce a well-formed half larva even if he made something or he manipulated the egg itself. Okay, so in the picture that you see here, this is the initial egg. So half of the egg has been destroyed with hot needle. So what happens is that, of course, half of the cell has been killed, and then the other one continued its development. So what happened is here, he still saw that it's possible to produce or a developed embryo. So next, we also have Hans Driesch. So Roos' fellow countryman repeated the experiment on sea urchin eggs, and the results was not the same as Roos' experiment. So he found a gastrula, only that it is small than the normal size, that developed into a whole and typical larva. Hence, the development process was known as regulation, which is the ability of the embryo to develop normally even when some portions are removed or rearranged. Okay? So come to think of it, from the experiment of Roux, he actually utilized a frog egg. So with his manipulations, he was only able to produce a half larva. But for Hans Driesch, he used a different model, which is the sea urchin. However, in his experiment, he was able to produce a whole typical larva. It's just that there's a difference with the size. Okay, so the results are very different or contrasted from Roux. Okay, hence he called it a regulation process. So that's how the mosaic and the regulative processes were actually debated upon. So dun pa lang makikita mo na one theory or one input cannot be true to all other organisms. So it always depends on what type of organism it is. Okay? So parang bumabalik tayo ngayon sa previous na mga theories na parang tama nga na nagkakapare-parehas lang talaga ang development ng isang organism kapag ka nasa embryological phase pa lang. But pagdating na sa mga more aggressive phases of growth, nagkakaroon na ng differentiation about the life cycle or the development. Okay? So in this picture, you will see the normal development of the sea urchin larva from two cell stage. So in Driesch experimentation, so once one of the cell has been separated, and of course it will die. So the cell that was left still developed into a full larva at the end. It's just that there's a discrepancy on the size. Okay? So, determinant pa rin talaga yung stage nung cell, okay, for the development. So, next is the discovery of induction. So, in here, the induction is actually a type of regulatory development. This is a process where one tissue directs the development of another tissue. And the classical experiment that has been devised for this discovery is by Spenman and Mangold in 1924. They grafted a dorsal lip of a blastopore of a newt amphibian embryo on a new site of another embryo. So, kumbaga, kumuha sila ng konting part nung dorsal lip ng isang amphibian embryo and tried to insert it to another site in the embryo. So, Spemann referred to the dorsal lip cells and their derivatives as the organizer. So, yung organizer na yon, sinasabi niya that it includes the notochord and the head of the endomesoderm. So, since it seemed to be ultimately responsible for the control of the organization and complete embryonic body. So, come to think of it, when you say notochord, that is the precursor of your nervous system. So, tama nga naman na ang notochord and the nervous system are the ones responsible for the control of the organization of the body. Okay? So, the Spemann organizer can induce the formation of an ectopic axis or twinned embryo. So, as you see here in the experiment, so this is the dorsal lip that has been removed from a Triton cristatus gastrula, and then they have inducted it or induced it to another part of a gastrula of the same species. So what happened is that a secondary embryo is induced. So if this is the first embryo that has been developed, the induced one is here inside. Okay? So you will see here that the pink picture was actually the first embryo, and then at the side of it is is the picture of the secondary induced embryo. Okay. So for the genetic studies, how was it commended? So this is because of the recollection of the Mendel's laws in the 1900s, which kick-started the studies involving genetics. So the genetic experiment Mendel did with his pea plants took him 8 years, and he published his results in 1865. So kung mapapansin nyo guys, yung mga concepts sa developmental biology ay patong-patong yung kanilang timeline. Okay? So in some parts of 1865, si Gregor Mendel, he already proposed some ideas about developmental biology, but then he focused on using pea plants. Okay? So during this time, Mendel grew over 10,000 pea plants, keeping track of the progeny number and type. So Mendel's work and his laws of inheritance were not appreciated in his time, but it was not until the 1900s, after the rediscovery of his laws, that his experimental results were actually understood. And he actually proposed the laws of inheritance. Okay, so dati hindi siya pinapansin, but then again, with the progress of science and of course researches and the application of genetic studies, kumbaga nahukay ulit yung mga dati niyang pinropose. Okay? So, Johann Gregor Mendel is actually the father of genetics. He carried out hybridization experiments designed to produce new color variants in pea plants, and he discovered that the fundamental laws of inheritance. He also deduced that genes come in pairs and are inherited as distinct units, one from each parent. And he tracked that the segregation of parental genes and their appearance in offspring as dominant or recessive traits. So, pag sinabi natin dominant, guys, ito yung mga characteristics na namamana natin. Na, for example, in a family, pag sinabing dominant, ito yung sa halos lahat ng family members, nakikita tong particular trait na to. So, for example, kulot na buhok. If the mother is straight-haired, and then the father is curly yung hair niya, and then the three offsprings are curly din ang buhok, it means that's an example of a dominant trait. Pero kung for example, may dalawang kulot, tapos may isang straight hair, yung straight hair, yun ang nagiging recessive. Okay? So, kung ano yung mas marami sa number, yun yung tinatawag nating dominant trait. Okay? So, he also recognized the mathematical patterns of inheritance from one generation to the next. Okay? So, in here, this is the results of the inheritance experiments of Gregor Mendel. So, Mendel's law of heredity includes 3 points or 3 laws. Okay, so the first law is the law of segregation. So each inherited trait is defined by a gene pair. Parental genes are randomly separated to sex cells so that sex cells contain only one gene of the pair. Offspring therefore inherit one genetic allele from each parent when sex cells unite in fertilization. Next is the law of independent assortment. So genes for different traits are sorted separately from one another so that the inheritance of one trait is not dependent on the inheritance of another. And then the law of dominance is an organism with alternate forms of gene will express the form that is dominant. So next we have the Danish botanist Wilhelm Johansen. So in 1909, he presented the concept that helped to link genetics and embryology by the distinction of genotype and the phenotype. Okay, so with genotype, this is the genetic information it acquires from its parent, controls development, environmental factors interacting with the genotype influence the phenotype. So when you say genotype, ito yung pinabasang characteristic ng inyong parents or basically they come from the genes. Okay? So kung yung gene ng mother mo and gene ng father mo, pag kinombine sila, that's what you call your genotype. Okay? Ang phenotype naman on the other hand is the visible appearance. So for example, pag kinombine yung gene ng mother mo at gene ng father mo, ano yung characteristic na magiging kakalabasan ng combination ng genes nila? So that's what you call your phenotype. So it's the internal structure and the biochemistry at any stage of development. So actually, the coming together of genetics and embryology was a slow and torturous process. And apparently, there are very little progress until the nature of the function of the genes were better much understood. So, it is also when the discovery in the 1940s that genes actually encode proteins that actually are the fundamental information that could actually help determine the stage of development. So in here, by controlling which proteins the cells or the genes are made of, so basically the genes are actually made up of proteins. And then itong mga proteins na to, they control the changes. So with what proteins that are visible in your body, they tend to signal behaviors, okay, that could intervene with development. Okay, so for the cell behaviors, um, this is actually the key link between gene expression and the development. Okay, so in brief, it is gene activity that gives the cell identity. So with with cell behaviors, we have the direction and number of cell divisions. Okay, so how many kumbaga divisions yung kayang gawin ng isang cell, 'di ba? 'Di ba nga katulad ng pinakita ko sa inyo kanina ng mga images, iba-ibang number or iba-ibang cell number stages ang pwedeng mangyari depending on what type of organism. And then of course, you also have the cell shape, the changes in the cell shape. So that's also one of the cell behaviors. Another one would be cell migration, kung saan ba sila pumupuntang part of the body. And then of course, the cell growth, how big or how small they can get. Of course, cell death, or commonly we call the apoptosis. Okay? So eventually, some of the cells in your body should die. Okay? Kasi kung hindi, Kapag hindi namamatay itong mga cells na to, what would happen is that, of course, you will be developing abnormal characteristics. Okay? Depending on what type of organisms. And then, you also have the changes in the composition of the cell membrane or the secreted products. Okay? So, genes control your cell behavior. So, how? So, by controlling which proteins are made by cell and when, and then whether a particular protein is synthesized in a cell requires first that its gene is switched on, that the gene is being transcribed into RNA, then translated into a protein. So in this one, we are already talking about the DNA being converted to RNA and then RNA to protein. Okay? So, kailangan switched on yung mga coding regions. And then, of a particular importance in development are genes that encode transcription factors or gene regulatory proteins, proteins that are involved in activating or repressing transcriptions. So, these transcription factors are very important because, kumbaga, they direct, okay, yung mga proteins na mas kailangan for development. And then, of course, if hindi naman kailangan or hindi naman necessary yung mga proteins for the development of a normal organism, they try to repress them. Okay? And then, transcription factors act by binding to the control regions of your genes or by interacting with other DNA binding proteins. So, the figure that you see here on the right side, so this is actually gene expression and the protein synthesis. So, a protein coding region comprises a stretch of a DNA that contains a coding region. So, this is your DNA. It has a coding region which contains the instructions for making your protein. Okay? And adjacent control regions. So this one, you have the promoter and your enhancer regions. So this is the enhancer region and then the promoter region. So this is at which the gene is switched on or off. So the promoter region is the site at which RNA polymerase binds and starts transcribing. And then the enhancer regions may be thousands of base pairs distant from the promoter. And transcription of the gene into RNA may either be stimulated or inhibited by transcription factors that bind to promoter and enhancer regions. So the mRNA formed by transcription is spliced to remove the introns, okay, and processed with the nucleus to produce mRNA that is exported to the cytoplasm. Okay, so may mga splicing pang magaganap. And then, in here, the control of the gene expression and protein synthesis occurs mainly at the level of transcription but can also occur at later stages. So for example, mRNA may be degraded before it can be translated. If not translated immediately, it may be stored in inactive form in the cytoplasm for translation at some later stage. And some proteins require post-translational modification to become highly biologically active. So, fina-follow lang natin yung mga processes ng ating DNA, from DNA to RNA, RNA to protein. Okay? So, we will not be we will be tackling more of this once we go further with our discussions. So for fate maps and cell lineages, so fate maps can be generated in several ways, and the technology has actually changed greatly over the past few years. So with the ability to follow cells with molecular dyes and computer imaging, it actually altered our understanding of the origins of several cell types. Okay, so if before we are just focusing more on the descriptive and anatomical studies for development, ngayon naman meron na tayong mga new technology na pwedeng i-adapt to check on fate maps and cell lineages. So, despite the different appearances of the adult animals, fate maps of the 4 embryos that you see here, you have the zebrafish, the frog, the mouse, and the chick. So it shows numerous similarities among their embryos. Okay? So basically, the fate maps of the vertebrates here that you see are early gastrula stage. So of course, the colors depict the different germ layers. Okay? So cell lineages is actually one of the most important programs of descriptive embryology, and fate maps constitute an important foundation for experimental embryology, providing researchers with information on which portions of the embryo normally become which larval or adult structures. Okay? So as you see in here, yung mga colors nga na makikita nyo, they do depict the different germ layers. And then, kapag compare nyo naman sa kanila, no, same kung ano yung mga nasa animal pole nila, 'di ba? Yun din yung makikita nyo dun sa iba. Okay? Minsan lang nga talaga nagkakaroon sila ng different arrangements like or different sizes katulad ng mouse and sa chick. Okay? So, E.G. Conklin in 1905, he actually tried to follow the fates of each early cell in the tunicate Stiella partita or the sea squirt. So in here, you see the fates of the individual cells that has been mapped by him. So embryos of this species many of the cells can be identified by their different colored cytoplasms. So the yellow cytoplasm that you see in here or on the pictures are actually marks the cells that form the trunk muscles. So A is actually a 8-cell stage. 2 of the 8 blastomeres contain this yellow cytoplasm. So ayan. So in B, this one, you see the early gastrula stage showing the yellow cytoplasm in the precursor of the trunk musculature. So from here, they eventually moved here at the sides. And then in the on the last picture, you see here now the early larval stage. So the yellow cytoplasm is already in the newly formed trunk muscles. And so that's how he traced it. So Vaught in 1929 actually adapted the use of dye marking. So he traced the fates of different areas of amphibian eggs by applying vital dyes to the region of interest. So what is important here is that the dyes or the dye stain cells do not kill them. So very safe siya for induction or para sa pagtuturok sa cell. And tinitignan niya sa microscope kung paano gumagalaw yung mga dye na yon. So in here you will see kung paano niya inintroduce yung dye. So dye size on the embryo are this one. So may darker and then may lighter dyes. So makikita niyo dito meron ng dorsal lip. So that's the where the cells begin to enter the embryo. And then next is here, kung makikita nyo, dorsal lip of the blastopore. So kung dito 6, 7, 8, 9, 10, 11, makikita mo habang pumapasok yung mga cells dun sa dorsal lip na yon, pumapasok din yung mga may dye. Okay? So nawala na dito yung 8, 9. No? So, nakapasok na sa loob. And then, eventually, pag tinignan yung section plane of the view, ayan na yung 1, 2, 3, 4. Tapos, sa lateral view, okay? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, he was able to map the position of these dye marks or the position of the movement of the development of the egg using the dye mark. Okay? So, one problem actually with vital dyes is that they become more diluted while um, there's a continuous cell division and they become difficult to detect. Okay? Kumbaga yung color ng mga dyes na yun, pumupo siya or naglalighten sila. Once once ko-continue yung progress ng cell division. So, mas nahihirapan ang mga scientists to check on the or to trace the movement or the pathway of the dyes. So, one solution that they have for this one is the use of the fluorescent dyes. So, these fluorescent dyes are not so intense that once injected into individual cells, they can still be detected in the progeny of these cells, even if there are or there has already been many divisions after the induction. Okay? So, in this one, you will be able to see the fate or the pathway of the dye using UV light. Okay? So, for example, in here, this is the fate mapping that for a certain egg of a zebra fish. So, if this is the animal pole at dyan nila nilagay yung dye, nung nag-develop na yung zebra fish, ito na yung kinalabasan or yung naging pathway kung saan pumunta yung mga cells na yon. Okay? So, if there will be overlapping colors, it will just indicate the cells from these regions are actually, kumbaga, or the cells of those regions are working together to form those organs. Okay? Or to form different regions of the zebrafish. So, one way of permanently marking cells and following their fates is to create embryos in which the same organism contains cells with different genetic constitutions. So, here, I present to you genetic labeling, and one of these examples or one of the techniques that is used for the construction of these genetic labelings are the use of the chimeric embryos. Okay? So, chimeric embryos are actually created from the same organism with different genetic constitutions for permanently marking cells and following their fates. So, embryos made from tissues of more than one genetic source, and this actually paved way for Mary Rawls to map the migration of the neural crest cells during vertebrate development. So, an example of these chimeric embryos are the chick and quail chimeras. So, basically, gumagamit sila ng tissues galing sa mga organisms which has one or more genetic which has more than one genetic source. So, magandang example yung chick at saka yung quail kasi 'di ba nasa isang family lang sila of birds. Okay? Also, they have used transgenic DNA chimeras. So, ito naman, the process is by transplanting cells from a genetically modified organism for animals that are difficult to meld a chimera from two species. And the gene is altered with the use of green fluorescent protein or GFP, a fluorescently active protein. Hence, the altered gene is called a transgene. Okay? So, if chimeric embryos do use natural genetic sources, for transgenic chimeras, they use modified or genetically modified organisms or GMOs. And they use the GFP for it. So such altered genes are what you call already a transgene. Okay? So for the application in or linkage of developmental biology with evolutionary embryology, even before Darwin, Charles Darwin, okay, J.V. Thomson demonstrated already that larval barnacles were almost identical to larval shrimp, and therefore he correctly counted barnacles as arthropods rather than mollusks. So it's also very important for the grouping or identification of um, the evolutionary family of such organisms. So another one, we have Alexander Kowalewski. He made the similar discovery that tunicate larvae had a notochord and pharyngeal pouches, and that this came from the same germ layers as those same structures in fish and chicks. So, brought about by Charles Darwin's evolutionary interpretation of von Bayer's law that relationships between groups can be established by finding common embryonic or larval forms. So he also argued that adaptations that depart from the type and allow an organism to survive in its particular environment develop late in the embryo. So there are two ways of looking at the descent with modifications. So first one is by pointing out embryonic similarities between two or more groups of animals. And then the second one could be the emphasis of the modifications to show how development has been altered to produce structures that enable animals to adapt to particular conditions. So, one of the most important distinctions made by evolutionary embryologists was the difference between analogy and homology. So, when you say homologous, structures whose underlying similarity arises from their being derived from a common ancestral structure, like what you see here on the image on the right. So, a human arm is homologous with a seal limb, homologous with a bird wing, and a bat wing. Okay, so all of these structures, as you see, they do have similarities. It's just that their differences are the shape and the sizes. Okay, but as you see, all of the structures do have humerus, they have the ulna, they have the radius, and then of course the hand, wrist, and the fingers. On the other hand, when you say analogous, those are whose similarity comes from their performing a similar function rather than their arising from a common ancestor. So for example, the wing of a butterfly and the wing of a bird are analogous with each other because they are used or they share the similar function for flying. Okay? So, next is for the application of medical embryology and teratology. So, um, teratology is actually the study of birth defects that can tell us how the human body is normally formed. And human infants are born with a readily observable anatomical abnormality. So some birth defects are produced by mutant genes or chromosomes, and some are produced by environmental factors that impede development. So for example, missing limbs, missing or extra digits, cleft palate, eyes that lack certain parts, or hearts that lack bulbs. Okay? So, genetic malformations and syndromes. So, malformations are abnormalities caused by genetic events like gene mutations, chromosomal aneuploidies, and translocations. And then when you say syndrome, it is a condition in which two or more malformations are expressed together. Like for example, the Holt-Oram syndrome or the mutations in the TBX5 gene. So TBX5 is actually a protein that is expressed in the developing heart and the developing hand and is important for normal growth and differentiation in both locations. So as you see, if a patient has this type of syndrome, meron siyang issues with the hands, and of course, meron din siyang issues with the heart. Okay? So, hindi normal yung growth ng mga hands nila and even the heart. Okay? So, next we have disruptions and teratogens. So, when you say disruptions, these are developmental abnormalities caused by exogenous agents like chemicals or viruses, radiation, and hyperthermia. And then you also have the teratogens, or in Greek, these are the monster formers. These are agents responsible for disruptions. And teratology is actually the study of how environmental agents disrupt normal development. So, substances that can actually cause birth defects include relatively common substances such as alcohol and retinoic acid. So, kapag ka nga, di ba, pregnant ang isang tao, usually, doctors do imply na wag iinom, wag gagamit ng mga pampaganda, usually kasi they, or these agents, or these teratogens, are actually contributing to the possibility of having birth defects. Okay? So, other chemicals also that may trigger birth defects can be heavy metals like mercury, lead, selenium, which can actually alter brain development. Okay? So, those are the basic information and the historical values and contributions of our philosophers in developmental biology. If you have other questions, please let us know so we could answer it. And if you have other concepts that you would like to clarify, you could use the following references. We have Gilbert and Baresi, 2016 Developmental Biology 11th edition. You can also read Papers of Love, Minelli, Stavelli, and the Book of Wolpert et al., which is the Principles of Development, 2nd edition. Thank you very much and have a nice day.